So thank you very much for the introduction, and thanks for the organizers for my opportunity to present here. I'm going to focus on a couple of concepts here. And yesterday we focused on the big picture and big data and how we think about activities of a large number of brain areas simultaneously, recorded either with unit firing or field potentials. And I'm going to focus on microcircuits as introduced by Alain. It turns out that there's very interesting neurobiology that's underlying the rhythm generation, and it's important to understand these neurobiological principles of what's happening in the microcircuit in order to understand what happens during these oscillations. And most importantly, in the context of future medicine, understanding how cells are interacting with each other through their synaptic connections opens up the possibility of very specific pharmacology to intervene in epilepsies. And I'll also talk a little bit about optogenetics, a very sort of experimental way that we can intervene in real time in the brain to prevent seizures, with the hope that this sort of real-time interventive approach could be ultimately developed for the treatment of epilepsy, but also other neurologic disorders. I also want people to interrupt me during the talk, so if there's anything that's not clear, please let me know. So I'm going to present work that was performed by a graduate student in the lab, Jordan Sorokin, a former postdoc, Jean Paz, who now has her own research laboratory at UCSF in San Francisco and the Gladstone Institutes, and Chris Mackinson, who is a current postdoctoral fellow in the laboratory. And all of this work was supported by the National Institutes of Health, especially NINDS, and a very good institution that supports research in epilepsy, the Cure Foundation. So this is an overview of today's talk. Can everybody hear me okay? It's okay in the back? Good. So I'm going to, this first part will be easy because Alain has already done a very nice introduction of the thalamocortical system and its roles in sleep and epilepsy. I'm going to focus more particularly on one particular component of this network, which is the reticular thalamus. The second part of my talk, I'm going to describe this new therapeutic intervention where we are going to change the mode of the thalamic cells to change them from one firing mode to another in real time in order to prevent seizures. And the last part of the talk, done by Chris Mackinson, is a mouse mutant model of epilepsy that's based on a mutation in sodium channels that causes a very specific defect in the thalamic circuit that predisposes it to epilepsy. And the insights that we've gained in that is which parts of the circuit are the most important in terms of regulating whether the seizures will happen or not. Okay. So what do we know about the basis of these? And it's been known for some time, as pointed out by Alain, that they may be related to the circuitry that's involved in the generation of sleep spindles, one of the characteristic rhythms that's generated during early stage sleep, slow wave sleep. So this is one patient where we have EEG records now from a sleeping patient, and then we'll see the same patient during the absence seizure. So this is a sleep spindle, as pointed out by Alain. This has this characteristic frequency of 12 to 14 hertz. And what you'll see is that it's present on multiple EEG electrodes. So it's a centrally generated rhythm. It can be seen all across the scalp, but it's not perfectly synchronized. So you note that the signal received on some electrodes is different than what's received on another. So there's an underlying rhythm generator which is recruiting erratically the different portions of the cortical network. Now this contrasts with what you see during the seizure. So this is now in the same patient. We've had to reduce the gain on the EEG or it would saturate. Now you can see these giant spike and wave discharges which are occurring at a slower frequency. And what you can see from this EEG activity is synchronization on a local level and on a global level. On a local level, you see that the amplitude of these events is much larger than the spindles, right? We're approaching 500 microvolts in amplitude out here on the scalp, several millimeters away from the brain itself. So that indicates massive synchronization locally between those two electrodes in the EEG. And also, if you look across electrodes, you're seeing the spikes occurring almost simultaneously all across the brain. So these are bilaterally synchronized. They're synchronized all across the brain during the spike and wave, which is quite different than what we see during the spindles. So the idea is there's a underlying rhythm generator that's similar for these two, but this is characterized by a massive increase in the synchronization. That's the theory. And there's evidence to support this. Alain pointed out some. This is another bit of evidence. This is from a cat model of absence epilepsy. This cat has been anesthetized with barbiturates, and so it's producing sleep spindles. These are recorded in the electrocorticogram. Here's the sleep spindle, which you can see on the power spectrum here, has a peak around 9 or 10 hertz. And then this animal is injected with penicillin, the antibiotic penicillin, which turns out to be, does anybody know what the neurobiological action of penicillin is? You've heard of penicillin though, right? Somebody knows? What is the neurobiological effect of penicillin? Yes, that's its antibacterial. It turns out it's a blocker of inhibition. It blocks GABA A receptors. So this was discovered, I think, in World War II when the neurosurgeons were in the field and they were treating soldiers that had received penetrating head wounds, and they would bathe the brain with a penicillin solution to prevent infection, and the soldiers would have seizures. So you don't apply penicillin to the brain directly. But in any case, if you give a very large dose of this intramuscularly, over time it gets into the brain in cats. And what you can see are these transition events where there is a slowing, as seen in the spectrogram. We're seeing the existence of spike and wave. These are larger amplitude, which is indicating increased synchronization. They're slower. And then you see events like this, which seem to start as a spindle and then transition to this spike and wave. So again, suggesting that the underlying circuitry for these two are related at least. So this nucleus shown here in this cross-section, this is a coronal section showing a particular potassium channel in the brain. This is KV 3.1. It's marking the thalamic reticular nucleus, which Alain referred to as the reticular nucleus, the RE. In the visual system, it's known as the parageniculate nucleus. And it is right here at the boundary of the thalamus. It's at the lateral extent and anterior extent of the thalamus. It sort of encases the thalamus, and it's all inhibitory cells. So it's a highly specialized nucleus that surrounds the thalamus, and it provides inhibition back to the thalamus. And this nucleus is the key to the rhythm generation in the brain. So understanding excitability in this nucleus turns out to be really important in understanding the seizures. These are just a couple neuroanatomical pictures of the reticular nucleus. Here is one thalamic reticular cell with its dendrites, and you can see they're splayed out across the thalamocortical projection. So the fibers that are interconnecting the cortex and the thalamus are all communicating with the reticular nucleus. So as it sits on that outer shell surrounding the thalamus, it's listening in on the thalamocortical dialogue. So these cells receive excitatory input from both the cortex and the thalamus. And in addition, they form a network among themselves. So this is now a parasagittal view along the edge of the thalamus, reticular nucleus, and you can see the cell bodies here marked with the stain nu-N, and in red is the calcium binding protein parvalbumin, which marks these cells really well, and you can see that they form networks among themselves. So they communicate between each other, and they also communicate back to the thalamus. And so now here's a cartoon showing you the thalamocortical circuit. And this is now a horizontal section through the rat brain, actually. And what you see here is the reticular nucleus, and I've drawn in a couple of neurons here. These are these inhibitory reticular neurons. This nucleus, unfortunately or fortunately, has many different nomenclatures. So I might refer to it as RT, RE, or NRT. They're all the reticular nucleus. So the inhibitory cells are sitting here in the reticular nucleus. They're in the middle of this thalamocortical pathway, and they provide inhibition back down to the thalamus. And so why are we so interested in this? It turns out that this thalamocortical circuit is what is responsible for the generation of that sleep spindle. And this is very well studied by Martin Deschênes and Mircea Steriad in the 80s, again in cats with intracellular studies. And I forgot to point out, all the insights that we've gained about this circuitry requires access to the inside of the cells. We have to be able to understand their synaptic inputs and what they do with those synaptic inputs, how they decide whether to fire or not. These sorts of understandings of the circuitry all require access to the inside of cells through intracellular recordings. So these are heroic experiments that were done in cat, recording from the thalamocortical cells, the TC cells, the reticular cells, the RT cells, or the cortical cells. And you can see they're all simultaneously active during the generation of that sleep spindle. So most noticeably, you see that the reticular nucleus tends to be firing repetitively almost throughout the entire duration of the spindle. You can see that it's burst after burst. Lots of activity in the reticular nucleus, which is simultaneously with the recorded EEG. But there's many interesting features of this network. One of them is this. So does anybody see a disconnect between the recording from a cortical cell and the spindle? It's pretty sparse, right? This particular neuron only fired 3 action potentials, participating maybe in 10% of the spindle. So remember, this is a cortically recorded spindle. It's recorded on the cortical surface. So what does that tell you about the nature of the spindle? It's subcortical, certainly, but what else? It's an emergent phenomenon, right? It takes the buildup of activity of multiple cortical cells to produce this. So presumably this cell is firing on these cycles, other cells would fire on different cycles. Together you would get this emergent network phenomenon observed in the EEG. And what you see in the thalamic cells during this is the same sort of sparseness, that this particular thalamocortical cell only fired twice during an individual spindle. So this is a kind of temporal sparseness, that individual cells are participating only occasionally during this activity. And again, for the network generation of the spindle that I showed you in the first slide of the EEG, other cells have to be participating at different times. Questions? So again, interesting neurobiology. This is one of my favorite slides because it makes the point very clearly that a neuron is not a neuron. And what we're looking at here is the expression of one particular type of excitable protein. This is a voltage-gated calcium channel, the T-type calcium channel that Alain mentioned in his talk. These are 3 members of the family, color-coded in yellow for CAV 3.1, red for 3.2, and blue for 3.3. And in this horizontal section of the rat brain, you can see here in the thalamus, it's bright yellow. So these thalamic relay cells all express very high levels of this one particular type of ion channel, the T-type calcium channel. You see some expression in cortex, not very much in striatum, not very much in cerebellum. And olfactory bulb, you see, interestingly, all 3. Shown here in blue, this is that reticular nucleus that I mentioned again. Those cells also express a member of this calcium channel. This is CAV 3.3. So both the types of thalamic cells, yes, Alain?
Is this layer 6 that is there in yellow?
Yes, that's layer 6, that's right. Yes, but whether it's enough to produce bursting is a different question. Yeah. But here you can see the uniformity, right? All of the dorsal thalamus, all the various sensory nuclei that are mediating sound, hearing, touch, vision, they all express very high levels of this. So this characterizes them. Thalamic cells just make a lot of this type of channel. And the reticular cells make an alternate version of the channel. And that turns out to be really important for this rhythm generation. So this was first described by Rodolfo Linhares and Henrik Janssen in the 80s. And what they showed, again with intracellular recordings, they stuck electrodes, sharp electrodes at that time, into thalamic cells and found this very strange form of electrogenesis. So these are now intracellular recordings. We're looking at the membrane potential of thalamic neurons. And when they injected a current in them to depolarize them, what they saw was this generator potential. You see this V-shaped, inverted V-shape that was produced in these cells, which was unique to thalamic cells. And what this generator potential does is it takes the membrane potential high enough that it starts to produce action potentials. So it's a boosting mechanism that produces action potentials in these cells. And Linhares and Janssen showed that it was calcium dependent because it was blocked by this divalent antagonist cobalt. And it was not sodium dependent because it was not blocked by TTX, the sodium channel toxin. So just to illustrate what happens when you express CAV3.1, the so-called T-type calcium channel. And this is just a simple demonstration of how you can get this mode switch of firing state of thalamic cells. And what we're doing now is making a recording from a thalamic relay cell. Remember, its basic job is to take sensory information from the periphery and relay it to the cortex. We've turned on a stimulus here and turned it off here. And what you see is that stimulus is creating a drive into these cells that's driving it above the threshold for sodium spike activation, shown here, the blue line. And the cell just continues to respond as long as that stimulus is applied. It's completely non-adapting. These cells don't do anything interesting. There's very little computation that happens at this synapse. It's basically taking that sensory information, transforming it linearly into a number of action potentials that get mediated upstream. Now, these cells are very interesting because they change their firing state as we fall asleep. And as we fall asleep, there was a withdrawal of neuromodulatory influences that tend to keep these cells depolarized. So this neuron is starting to fall asleep. You see I've moved its membrane potential down a little bit by 5 millivolts. Now that same sensory stimulus is producing fewer action potentials. So it's less responsive, and so that sensory information now is less effective at getting to the cortex, right? So the cortex is starting to ignore what's going on in the outside world. And as we fall further asleep, you see now this stimulus becomes subthreshold. So it's completely ineffective in producing activation of the cortical part of the circuit through this relay. But these cells have this strange form of activity, this paradoxical excitability, as we make them even more hyperpolarized. And here you can see the start of this generator potential. There's this little triangular wave that's starting to develop at this particular degree of hyperpolarization, and it gets fully expressed when we're fully asleep. Now the membrane potential is very deep, around minus 80 millivolts or so. This same now sensory stimulus produces a very strong burst of action potentials. So you can see repetitive firing that lasts as long as the generator potential lasts. And it's a really striking change in phenotype, because look at the frequency that we're seeing here. It's on the order of several hundred hertz. This highly clustered set of action potentials, which is producing then very strong output to its cortical partner, because you see the summation of all the synaptic outputs that would be produced there. So this change in firing rate, or firing mode, is critically involved in the generation of this rhythms, and that's was described by Lan, and I'll just make the point here. This internal thalamic oscillatory network is well preserved in brain slices from mouse, cats, ferrets, and rats. And it's different orientations, but all you need to maintain the thalamic recurrent circuitry is to keep this axon intact and this axon intact. So we have excitation from the thalamocortical cells to the reticular cells, and then feedback inhibition from the reticular cells back to the thalamocortical cells. In rodents, in the somatosensory cortex, we can obtain this with a horizontal section through the brain. And in the ferret, it's a parasagittal section. So you just need to, as Alon said, you need to section the brain in the right direction, and then you can retain this connectivity. And yes?
Do you know if anybody tried on a cat? Cat slice on the thalamus?
I think doesn't. I don't know for sure. Yeah.
I never heard about it.
I would imagine it would probably work with the LGN. Yeah. Okay. And what we and other laboratories have done is exploited the fact that we have this internal thalamic network that's a very potent rhythm generator to study rhythm generation as is relevant to the circuit. And like this is cartoon form now showing how these two types of cells interact to produce the oscillation. Output from the reticular cells. So remember, they express calcium channel CAV 3.3, which was the blue one in that illustration. They fire a burst of action potentials that causes release of GABA at this synapse right here to activate GABA A and some GABA B receptors. And this membrane hyperpolarization that's been produced by the iPSP is what primes the calcium channels and allows these cells to fire this post-inhibitory rebound, PIR, which is the characteristic feature of these TC cells because they express that CAV 3.1 channel. So these cells, when they fire, they produce many action potentials, which causes the release of glutamate within this intrathalamic circuit to reactivate the reticular cells, and then the oscillations will just continue. Now, we showed many years ago that one of the drugs that's used in the treatment of absence epilepsy is ethosuximide. And ethosuximide turns out to be an antagonist of the CAV3 channels. And this makes sense. I mean, I should say that in terms of precision medicine, anti-epileptic drugs are not very precise. They're classic dirty drugs where each of the drugs that's used, phenytoin, ethosuximide, et cetera, has been shown to have many cellular actions on ion channels, synaptic transmission, et cetera. But one of the effects of ethosuximide is to block those calcium channels. And that makes sense in this context of rhythm generation. If you can reduce the ability of these relay cells to fire post-inhibitory rebound, the thalamic portion of the thalamocortical loop is going to be less able to generate these rhythms. But this was an idea that we developed in brain slices in rodents, and so it was never really tested. And now we have the possibility to test that. So that causes me to switch to the second part of the talk, and how we're going to use optogenetics, this way to modify cells to render them sensitive to light, so that we can change their firing mode to test this hypothesis that this burst firing mode is very important in generating the seizures. So how many people here have not heard of optogenetics? Everybody has. You have not heard. Okay, a couple. So optogenetics is the idea of using gene therapy, so using a viral approach to bring in some DNA into neurons and cause them to express proteins that they don't normally express. Okay? It's a very powerful approach. It might be useful in treatment of human diseases. There's actual clinical trials that are starting to use gene therapy. Optogenetics takes the idea that you take a protein that's not even a mammalian protein. This is a protein that comes from bacteria. It's a light-sensitive protein called channelopathies. You take the DNA for that and put it in a virus and then inject it into the brain, and neurons now start to produce this protein, right? So it's very strange because it's not something they normally express. But the protein then is made by the cells because it's been tricked by this viral gene therapy approach. And this protein makes it out to the membrane. And the protein is channelopathies. It's actually a pore that lets ions in. And so it's almost like an excitatory neurotransmitter. It lets ions in and out of the cells the same way that a glutamate receptor does to produce excitation. And this protein can be controlled by the application of light. It's a light-sensitive protein. Channelopathies itself is activated by blue light. And what's very nice about this is it's temporally precise. You can turn on and off a laser onto neurons and very rapidly open these channels and close them. And that turns out to be therapeutically quite advantageous for thinking about intervening in brains during certain states. So there's two main opsins that have been developed. These are the two prototype opsins. Channelopathy version 2, which is still highly used. It's activated by blue light. It sits in the neural membrane like an ion channel. And when it's activated by blue light, it allows sodium ions to flow in, just exactly the same as a glutamate receptor. And that causes activation of neurons. The second opsin that's used is halo-rhodopsin, abbreviated NPHR, and this causes activation of a chloride ion pump. And so it has the opposite effect. It's pumping negative ions into the cell, and that tends to hyperpolarize them to suppress activity. So we can inhibit cells or activate cells by these two opsins. And importantly, they have different spectral characteristics. So you can apply blue light to activate the cell or yellow light to inactivate the cell, and you can express both of these proteins in the same cell. So you can bidirectionally, in real time, turn on and off neurons. It's very exciting. We hope that it might ultimately be useful in patients, but that's going to require a lot of optimization in terms of gene therapy, et cetera. So it's the important things are that it's can be used in real time to modify the activity of cells. And so here's a recording from the first description of halo-rhodopsin. You can see channelopathy is reliably causing activation of action potentials. Hold on just a second. And then halo-rhodopsin is suppressing that activity. Yes?
Can these interventions be neurotoxic?
That's a good question. Can I answer that at the end in the discussion period? It's actually a very hot topic, is what happens when you express foreign proteins in neurons. So there is the possibility, but I'll get back to that at the end. Okay. So now we're going to talk about the effects of halo-rhodopsin on thalamocortical cells. And in a previous study where we were intervening in stroke-induced seizures, we found that if we expressed halo-rhodopsin in those thalamocortical relay cells and activated by yellow light, we could produce this membrane hyperpolarization. These are now intracellular recordings just showing the robustness of this optogenetic signal. So yellow light, yellow light, yellow light, it's a train of stimuli. In the blue traces, we're recording the direct response, the channels opening and closing in a voltage clamp recording. And what you can see is it's completely reliable. Every time we turn on the light, channels open and they close. They open and they close. And if we then switch the amplifier so we allow this cell to fire in so-called current clamp mode, now you can see that every time we activate the halo-rhodopsin, the membrane potential gets pushed down. And just like the synaptic response, synaptic inhibition, when this decays, this results in the activation of a post-inhibitory rebound burst. But there's one key difference, and that is that there's no plasticity in the optogenetic signal. There's no short-term plasticity. So every time we apply light, we're pushing the cells down just as effectively. And so this would be an extremely powerful way to synchronize the network, right? Because we're pushing relay cells down at the same time. They're going to tend to fire a rebound response at the same time. And we hypothesize that this would be a sufficient signal to initiate a seizure. And so that's what we tested in the first part of the experiment. And so here's an experiment I'm showing you now, EEG recordings from a rodent model of epilepsy. This is the stargazer model of mouse absence epilepsy. I'll show you a video in a second where you can see they have the same absence phenotype as humans. Here we're applying an 8 Hertz light train, and this is the frequency of the seizures in the stargazer mice. And what we're doing is we're hyperpolarizing the relay cells periodically at 8 Hertz unilaterally. And during this train stimulation, you see we activate the ipsilateral cortex, but very rapidly, within less than a second, we're also generalizing the activity. You can see this strong spike and wave activity in the contralateral cortex. So driving burst firing in relay cells is a very effective way at producing seizures. And if you look at the spectrogram, you can see that it's almost exactly overlapping with naturally occurring seizures in these animals. There's one small induced peak, which is our stimulation frequency. So maybe that's not surprising because this is an epileptic animal, but we also showed that we could apply very brief stimuli, 1 second of this light train. And then more recently, we've started to include multi-unit recordings. And so the approach is a combined optical and electrical approach where we put an optrode deep into the thalamus. We're delivering light to the thalamus at the same time as using single wires to pick up unit firing. So we're looking at action potential firing in the thalamus during these stimuli. So shown here in the red circles are single action potentials. And then in green here are the blocks that indicate bursts. So these are periods of activity where it looks like the neurons are firing these strong post-inhibitory rebounds. And you see, before the seizure, there's no bursting. We start applying the yellow light stimuli, and very rapidly, we recruit burst firing down in the thalamus. And then also very rapidly, this results in recruitment of this bilaterally synchronous discharge. And so you see huge increases in power, especially in this theta prime frequency range, which is 7 to 10 hertz. So these driving pulses, which are actually producing periodic rebound inhibition, are extremely effective at inducing seizures. And this is the most striking result. So now this is a different animal model. It's another animal model in rats. This is the Wag-Rai rat. Based on a Wistar rat. Again, we have a halorhodopsin expressed in relay cells. And here is a simple, one single light pulse applied into the thalamus. And you see how this results in the generation of this widespread synchronous activity. And when you look at the multi-unit activity, it's even more striking. So here's pre-seizure activity. The X's here are showing single spiking, single spiking. Here is the one stimulus. And if you look at the multi-unit activity, here is this population burst. A small group of relay cells are firing burst responses. The light is turned off at this point, and this single event is enough to recruit the larger network into the seizure that occurs with repeated bursting and bilateral synchronization. There's a bunch of controls you can do for this. You can do light controls without virus. Here is a virus that just expresses a carrier fluorescent protein, so it's still a viral transfection to test for the potential neurotoxic effects. Light produces no effect in those cases. And as I said, we can do this in mouse and rats. So that was a simple proof of principle. If we start driving bursting in thalamic cells, can we initiate a seizure? But more relevant for therapy is, can we block the burst? Once the cells stop bursting, if we block the bursting, can we abort the seizure? And for that, we used a second generation opsin, which is called SSFO, stabilized step function opsin. And as its name indicates, it has this interesting feature that when turned on by blue light, just like channelopathies, it turns on, but it stays on. It's a bistable opsin, so that it goes into this conductive mode as soon as the blue light is turned on, and it'll stay on for 20 or 30 minutes. It's a very long-lasting switch in the permeability of this molecule. Interestingly, it's also bistable in that its photocycle can be exploited to turn the opsin back off again. So application of yellow or amber light will turn off this opsin, and so we can switch it up and switch it down by applying blue or yellow light. And one of the interesting features of this opsin is, you can see as it was characterized in these recordings from pyramidal cells, is it doesn't produce a super strong activation itself. So here's blue light being activated. The cell doesn't really have very much firing induced by this depolarization. So it's simply modulating the sensitivity of these cells by producing this slight depolarization. So now incoming synaptic inputs would be more likely to activate action potentials, but the light itself is not actually driving the cells to fire. So this is a perfect opsin for us for our studies of thalamic bursting, because I've showed you in the slide that as the cells are depolarized, they're incapable of bursting. So the question is, can we detect a seizure, switch the mode of the cells from burst firing to non-burst firing by application of activation of the SSFO, and will that suppress the seizures? And sure enough, it's extremely effective. So this is now the same presentation I showed you earlier. Single X's here are action potentials that are detected on this multi-electrode recording. Here you can see the seizure starting to develop. You see bursts occurring in these thalamic electrodes. So grouped activity, grouped action potentials. Here is the cortical surface EEG on the ipsilateral side. You see it starting to develop into a seizure. And at this point, we apply blue light, and that turns on SSFO, and it pushes the membrane potential of the relay cells up by a little bit, by 5 or 10 millivolts. It changes their firing mode. Now you see there's no green here at all. We've prevented these cells from bursting by depolarizing them, and what we get is an instant normalization of the EEG. So the seizure is aborted by suddenly changing the firing mode of these cells. And this is just showing the spiking activity of these cells. You can see the blue light is turned on here. This is repeated raster trials, as Alain described. The opsin turns on, the cells increase their spiking, and then the yellow light turns them back off again. It's quite repeatable from trial to trial. Again, we have appropriate controls for this. So if we apply the blue light in animals that have another virus injected into their thalamic relay cells, just expressing the fluorescent protein, there's no effect of the light itself, and the seizures are unaffected. And I think that we have some statistics. And so what's shown here in blue are sham stimuli. And so these are stimuli that would come on randomly that would not turn on the light. And this is to account for the fact that these animals were naturally having some seizures themselves. And so we needed some prediction that the effects we were seeing were not due to the spontaneous occurrence of the seizures. So you can see that the number of action potentials goes way up when we turn on the SSFO. So we pushed them into this tonic firing mode. They're not firing bursts. The bursts are completely eliminated in this post-burst period. And this is associated with a blockade of the seizures. So again, this is now a sham stimulus and stargazer. So here's the detected seizure. So we use a very simple detection method, just looking at the line length of the recorded EEG signal. We can detect it relatively rapidly, usually within a second or two of its onset. Here's a detected seizure where we didn't apply any stimulus, but it's a sham manipulation, and you see the seizure continue. But here's a case where the seizure was detected, the blue light came on, and the seizure is instantly aborted. The statistics show that when we do this in many animals across many trials, we reduce the duration of the seizures when we switch their mode out of burst firing mode with this SSFO activation. So the sham stimuli are shown in blue, and you can see this leftward shift on both the contralateral and the ipsilateral side. So briefer duration of seizures when they're interrupted by the SSFO in both the stargazer mouse and the wag-rye rats. So therapeutically, we're able to detect the seizures and shut them down in real time. So it's a really exciting possibility for future therapies. And then here's the controls in the case where we just use the virus without the opsin, showing no effect on seizure duration. So to conclude from this part, burst firing in the thalamic neurons is required for absence generation. It's both necessary and sufficient. If we drive burst firing in a group of cells with this grouped hyperpolarization and rebound bursting, it's sufficient to initiate a seizure. And if we block the burst firing with SSFO, the seizures are blocked. So this suggests to us that targeted CAV 3.1 inactivation perhaps in real time would be the best type of treatment for absence epilepsy, to block the ability of these cells to fire their bursts, especially in the context of a seizure. There's going to be some challenges to develop that, which we can talk about during the discussion. Okay. In the last part of the talk, I'm going to talk about our most recent studies of thalamocortical seizures in a spontaneously occurring mouse model. And this now has to do not with calcium channels, but with sodium channels. So you all know sodium channels are the biological basis of excitability in the brain. Sodium channels are what allow action potentials to occur. There's several different members of the sodium channel gene family, SCN1 through 11. Not all are expressed in the brain, but you see that all that are expressed in the brain, 1A, 2A, 3A, and 8A, are all associated with epilepsy. So if you alter the activity of this basic excitability of the brain, it will either induce reduced susceptibility to seizures, or more commonly, actual epilepsy. So sodium channels you don't want to mess with. And you can see there's various types of seizures that are associated with these different channels. There's a syndrome called Dravet associated with NAV 1.1, which is 1A. Also with 1.2. There's a different form of epilepsy called Jeff's plus, generalized epilepsy, and febrile seizures plus, benign familial neonatal infantile seizures. And the one I'm going to talk about is SCN8A, which confusingly corresponds to NAV 1.6. So the protein doesn't align exactly with the numbering of the gene for this particular family. This mutation, where a gain of function, if you start increasing the function of this particular sodium channels, it leads to epileptic encephalopathy. But in loss of this protein can actually be protective and can lead to absence epilepsy. And that's what I'm going to talk about now. So again, SCN8A, the gene, encodes NAV 1.6. Gain of functions mutations lead to this severe form of epilepsy. These mice, the so-called MED mice, are a spontaneous mutation that results in a heterozygous deletion of this of the SCN8A channel. So this is a loss of function of half of the gene dosage for this particular ion channel. And Chris Mackinson in the lab was a graduate student of Andrew Skye at Emory, and they were studying what the effect of loss of 8A would have on seizure susceptibility. And they did this in a convulsant model. So this is seizures induced by flurothyl. And this is a seizure threshold test where they're dripping flurothyl into the chamber and measuring the latency to produce myoclonic jerks, MJ, or generalized tonic-clonic seizures, GTCS. And they use this clever genetic approach to try to modify sodium channels in different types of cells. And so they looked broadly at cortical cells with this promoter, FOXG1, across the MED animals with these animals in a Kree-Lox approach. And what they showed is an increased latency. So these animals were actually less likely to have seizures if they're missing one of their sodium channels. Okay? And then they further refined this by using different approaches. So EMX1 and CAM kinase 2 both target excitatory cells specifically. And what they found is they have the same effect as broad cortical suppression. So if you take away the NAV 1.6 from cortical excitatory cells, you reduce the likelihood of producing seizures. But there was little effect or no effect when they did this in mouse lines that would reduce expression in interneurons. So this is the broad promoter, DLX56, and this is a more specific promoter which affects many inhibitory areas, but not the thalamus. And then what they found was this very, very interesting phenotype, interesting to me, that if they looked at the expression of absence seizures, so these are again rodent absence seizures, they're a little faster than in humans, but they're spike and wave. They go about 8 hertz or so. And what they found is if they broadly inactivated one allele of the 8A channel in interneurons with this DLX56 promoter, they saw the occurrence of many, many spontaneous absence seizures. So loss of function of a sodium channel is actually leading to an epilepsy phenotype. Complete opposite of what they showed with the seizure susceptibility of the excitatory cell. And then they did not see this phenotype when they used the PP1R2 promoter. And that was interesting because this promoter fails to influence the expression in inhibitory cells in the thalamus. So this result between these two really makes it seem like it's changes in excitability in the reticular nucleus, those inhibitory cells in the thalamus that are responsible for this epilepsy. And that's what Chris came to my lab to try to understand. So here's first the electrical phenotype of these animals. They have exactly the same type of seizures that I showed you in Stargazer and Wag-Rai and people. They have spike and wave seizures associated with these behavioral absences. You can see them in thalamic recordings and cortical recordings. The spectrogram has this beautiful 8 hertz oscillation, and you can see the harmonics, which are quite indicative of spike and waves. You get these higher frequency components that are associated with the spikes and the spike and wave, and it's absolutely what human absence epilepsy looks like. This also has this characteristic slowing during the time course of the seizure, which is exactly the same phenotype you see in humans. It starts at a high frequency and ends a little bit lower, like 10 or 20% lower in frequency. This, by the way, may have something to do with the termination of the seizures. As they get slower, they may be less capable of sustaining themselves. So what's going on in these mice? And first question we asked is the thalamus affected, right? We have this genetic evidence that this was the site of activity of the mutation, and we tested this now in our brain slices. So this is that same horizontal brain slice method that I described before. Here's a brain slice, here's the thalamocortical cells, the reticular nucleus, and a stimulating electrode that we've placed in the internal capsule. And this stimulating electrode will activate cortical thalamic outputs and thalamocortical outputs. Both of those types of excitatory connections will activate the reticular cells. Remember, it's listening into that thalamocortical dialogue. So we can mimic a cortical response by stimulating here. And what that does is it initiates activity in this recurrent thalamic loop. And in wild-type slices, we can get activity that goes on for a couple of cycles, so we get a weak oscillatory response. But in the 8A animals, you see you get this very much longer-lasting response. It's what we would call epileptiform, right? It's this self-sustained electrical activity that goes on for many seconds, the same sort of time frame that we know occurs in the seizures in these animals, and the same characteristic frequency. You can see in the autocorrelogram of the spike meter output, you get this beautiful oscillation in the wild-type slices that's absent in the, that's present in the 8A slices and absent in the wild-type slices. So yes, there is something going on in the thalamus of these animals. And then Chris looked at the anatomy, and I hope you can appreciate this. This is now looking at the location of the 8A protein in different neurons in different cortical areas. So this is immunostaining for that 8A protein. And you can see these beautiful segments in the cortex. So these are corresponding to the initial axon initial segment, which is where the axon first leaves the pyramidal neurons in the cortex. And so there is high expression of 8A in that part of those cells. If he tried to find out expression of 8A in the cortical cells, so this is now expression for GAD65, 67, marking inhibitory cells, there's no co-localization of 8A at all. So it looks like the 8A is really affecting excitatory cells primarily, and this can explain the reduced seizure susceptibility in those animals. By contrast, when he looked down in RT, here's an axon segment, right? It has this same shape. It corresponds with ankyrin staining, not shown here, which is a marker of axons. But here in the reticular nucleus, you see this 8A staining corresponds to the GABA staining. You can see in yellow here. So there's many of these segments in the reticular nucleus that are axon-like segments. And so it looks like 8A has a specific role in mediating the activation of reticular cells, these inhibitory cells that are in the thalamus, but not so much of a role in the inhibitory cells in cortex. Okay, so that leads to the question, would inactivation of 8A in the thalamus, if we just reduced the expression of this protein in the thalamus, would that then be sufficient to produce epilepsy? And so they used a molecular genetic approach called knockdown. They used short hairpin RNAi to interfere with the RNA for 8A when expressed in the thalamus, and they expressed it either here where it overlaps with the reticular nucleus, which you can see in red, or they expressed it more medially, so there was little expression in the reticular nucleus. And only when they when this gene approach to modify 8A overlapped with the reticular nucleus did they see expression of seizures. So this is sufficient. All you need to do is reduce the expression of one sodium channel in one brain region. Is it sufficient to induce seizures? And this is really the first demonstration of a thalamic origin for thalamocortical epilepsies. As Alain said, there's much evidence that the cortical hyperactivation is often a initiating event in the generation of seizures. Here, this shows that that specific deletion of one particular protein really affects the circuitry in the thalamus in a way that can produce thalamically initiated seizures. So how am I doing on time? I'm just about done. Okay. So I will skip a couple of parts here where we looked at some detailed intracellular physiology to look at the effect of 8A, and just tell you that we saw some changes in their ability of the cells to fire tonic action potentials and burst action potentials. That it can explain in part the epilepsy, but we really wanted to know how effectively these inhibitory cells were producing their inhibitory output. And for that, we went back to the optogenetic approach. So one very nice feature of optogenetics is you can use it for circuit busting, which means you can directly activate one group of cells, record their synaptic response, and understand what are the features of that synaptic response. So this is the experimental paradigm for testing that. We know that the reticular cells are affected, and we know that if you take away 8A channels from the reticular neurons, that's sufficient to produce epilepsy. What we wanted to know was how effectively they inhibited their targets. So how effectively do they inhibit the relay cells, and how effectively do they inhibit each other? I haven't said very much about this, but our working model is this network of inhibitory cells that I showed you in one of the first slides, where they're interacting with each other. This, we think, is a protective mechanism in the thalamus. The fact that the reticular neurons are inhibiting each other would tend to prevent synchronization in the network. When these cells are active, they're going to inhibit their neighbors, and they're going to prevent the synchronous activation that we think is characteristic of the seizures. So if this synapse becomes more effective, which it does in the presence of benzodiazepine treatments, or if it becomes less effective in the context of this gene mutation, this is going to increase or decrease the susceptibility to seizures. And what you can do with optogenetics is use virus to express channelopathies in the relay cells, and it gets expressed all in the axons, all the way out to the termini. So that means you can use light to directly evoke release of GABA from this synapse and this synapse. It's a very powerful approach, because now we're saying, okay, what is the effect of this synapse on this cell, or the effect of this synapse on that cell? So Chris did this, and he looked at the output of reticular cells onto thalamocortical cells. So remember, this is the output that's driving the oscillation. When the TC cells receive this inhibitory input, they get pushed down and they fire the post-inhibitory rebound burst. And what he showed, so this is now application of a light train at 20 hertz, repetitive blue light pulses, So we're evoking GABA release from those synapses. We get some short-term plasticity. The first response is large, and the secondary responses get progressively smaller and smaller. What's shown in different colors are different trials, and you see it's quite repeatable. Every single time we apply these trains, you get this characteristic reduction in amplitude. And eventually, once you start driving this at high frequencies, you start seeing synaptic failures where some of the light pulses will fail to produce a synaptic response. And when you characterize this, you look at the ability of this synapse to fire as a function of frequency. It's unaffected by this mutation. So think about this. These are the axons of reticular cells. They're missing some of their sodium channels, but at least for this synapse, their ability to fire these at these different frequencies is unaffected. So this part of the network that's driving the oscillation is intact. On the other hand, when he looked at the intrathalamic network, intrareticular network, the connections between the reticular cells, he saw many failures, and they're marked with red squares here. So here's another stimulus train. You can see here's a response, here's a response. There should have been a response right here, but there was none. That's a failure. There's another failure. There's a couple more failures. So this part of the network is more fragile. This part of the network, which we think is the most important one in regulating the synchrony in the network, is fragile, and it's more fragile in the presence of this loss of 8A. So you can see here's repeated trials in different colors, and you can see on no individual trial was it 100% successful. It's stochastic. Here on the purple trace, it responded here, 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 and here, but not through here and here. And here, the green trace, you see a different outcome. But overall, in the 8A, they were much less able to sustain this activity. And at 10 hertz, you can really see this difference. This is now the number of failures. So how successful was that synapse? The thalamocortical cell synapse you see is almost completely effective. It's a failure rate of 5% or less at 10 hertz, independent of whether the sodium channel is mutated. But here at the reticular nucleus, we have very low failure rate in the wild type, but in the presence of this hemizygous deletion of 8A, the loss of sodium channels from that part of the axon, now we get a very high failure rate on the order of 50%. So these cells have lost the ability to inhibit each other, especially under these conditions of repetitive activation, exactly the conditions that happen during a seizure. The synapse is being activated over and over again, and now it's failing when you need it to be acting most effectively. So let me finish up really quickly so we have time for one or two questions. The first part, I said burst firing really is a critical component, and it's going to be an important for current and future therapies, we think. And on the second part, this RT output to other RE cells, other RT cells, is really a critical control point for the seizures. So just to acknowledge, again, this is Jean Paz that did most of this work, and Chris Mackinson and Jordan, and our collaborators, Carl, who provided the optogenetic tools, and then Eric, who helped with some of the EEG analysis. So I have time for a couple of questions, I think.